Thank you, and thanks so much to ACS AGRO for the opportunity to reach even more people and share more information about insects with even more of the public. So, according to the internet, it's amazing what plants can do. In fact, it looks like there's nothing plants can't do. Just a brief survey shows that herbal teas can enhance immunity, boost brain function, combat exhaustion, and spices can boost your brain power. Burdock root can cure cancer, and my personal favorite, papaya seeds have amazing unknown power. Nothing more impressive than unknown power. In fact, some of the claims on the internet kind of defy belief. I don't know how you're accustomed to thinking of celery, but it's kind of a shock to think of it in the context of being the vegetable Viagra. That celery is a sex magnet, and people who want to be babe magnets should try munching sticks of celery, says a top Swedish sexologist. Advice here, to stalk up on celery for better sex and less stress. Kind of makes you want to rethink your tuna salad recipe. Well, in fact, plants really do have some remarkable physiological activities, and they have these activities because they produce a staggering diversity of phytochemicals. So phytochemicals are now being promoted as substances that improve human health. The phytochemicals are, in reality, those chemicals made by plants that have no direct contribution, that make no direct contribution to photosynthesis and other essential plant functions. Phytochemicals are characteristically very idiosyncratic in their structure, diversity, and distribution among plant families. Just to take one plant with very pronounced and well-known physiological effects, Cannabis sativa, has a remarkably large inventory of phytochemicals. Over almost 500 have been identified in all kinds of structural classes, and that's just one plant. And phytochemicals are, by their very nature, biologically active, and this biological activity manifests itself in all kinds of ways. Many phytochemicals are antimicrobial. They can act as hormones. They can induce enzymes. They have antioxidant activities. They can interfere with the bioavailability of nutrients. There are all kinds of activities that have been demonstrated to be possessed by phytochemicals. So some of these biological activities in humans happen to promote health. Some are familiar. Sulforaphanes, for example, in broccoli, cabbage, and the other cruciferous vegetables are known to be associated with a lower risk of all kinds of forms of cancer. Carotenes, precursors from vitamin A, also have beneficial effects relative to reducing the risk of cancer. Many monoterpenes are also demonstrably health promoting. But there is a dark side to phytochemicals, and people encounter this dark side when they use or abuse various and sundry herbal products. Probably most famously in the last couple of years, about 2 years ago, Lamar Odom, a Kardashian associate, ended up hospitalized probably through abuse of what are called herbal erotic aids, so an herbal form of Viagra that is used to treat erectile dysfunction. And this is not an isolated case. Many herbal products are known to be toxic, and there are regularly reports in the newspaper, accounts of people who are inadvertently poisoned or even killed by consuming the wrong herbal product. This was a story that just appeared a couple of days ago. 2 people poisoned after drinking herbal tea from San Francisco's Chinatown. Turns out that the tea that they drank, which they purchased at a store, contained a poison called aconite, which is derived from aconitum, which is a plant that has, among other names, queen of all poisons. So you would think people would maybe think twice about consuming an herb known as queen of all poisons. Well, the thing about phytochemicals, and in fact any toxins, is, as Paracelsus said in 1538, the dose makes the poison. So there are phytochemicals that may have beneficial effects in low concentrations, but may cause toxicity or even fatalities at higher concentrations. And this includes not just nutrients, but also many of these herbal products that are rich in phytochemicals. Well, plants don't make phytochemicals to function as altruistic drugstores for humans. By and large, phytochemicals are produced by plants to mediate interactions with other organisms. So plants make repellents and poisons for dealing with their enemies because they can't run away from them, being firmly rooted in the ground. And they also make phytochemicals as attractants and rewards for their mutualistic partners, such as pollinators. So of necessity, because these chemicals are designed to interact with other living organisms, they have biological activity. Essentially, all plant consumers have to deal with phytochemicals, and some of them are quite pronounced in their biological activity. Just case in point, Nicotiana tabacum, the tobacco plant, famously contains the alkaloid called nicotine. Nicotine is able to interact with what are called nicotinic receptors within the nervous system in nerve cells. So normally respond to acetylcholine, which is a neurotransmitter substance that elicits a neurological activity. Nicotine gets in there and causes nerves to fire inappropriately. So it is essentially a neurotoxin. Now, humans have known about the neurotoxic properties of tobacco for a very long time, and in fact, even before Europeans discovered the fumitory pleasures of consuming tobacco, i.e. using it to smoke, it was used as an insecticide. In fact, that use continues today. Blackleaf 40 is essentially a nicotine solution that's used to control insect pests on plants, particularly aphids and other such pests. And as you can see from the warning label, it's very toxic. Now, we humans are at reduced risk of toxicity from ingesting phytochemicals because we feed very broadly. We have an astonishingly broad diet. In fact, if depending on your preferences in pizza toppings, you could eat representatives of 4 different kingdoms. If you like, say, cheese and pepperoni and mushrooms on your pizza. So our broad diet minimizes our exposure to any particular type of poison. As well, we humans are long-lived, large, very mobile. We can remember adverse effects, and we can communicate with each other to avoid exposure, minimize our ingestion of toxic substances. Witness the news stories about not going and ordering Wolf's Bane or Queen of All Poisons tea. As well, the capacity of humans to process and ingest plant poisons seems almost unlimited. You can see the many varied ways humans have come up with to enjoy the neurological activity of nicotine without actually dying, at least immediately. Now, caterpillars don't have those options. They're not large, mobile, long-lived, and really they don't have much in the brain department. You can just see from the relative size of their head and their guts that it takes a lot of guts to be an insect herbivore, but not necessarily a lot of brains. The tobacco hornworm, as the name suggests, is a specialist that feeds exclusively on plants in the tobacco family, the Solanaceae. And in fact, it is routinely found consuming large quantities of the foliage of tobacco, which contains nicotine. And they ingest enormous quantities of nicotine. They can increase in size 10,000-fold in just a week. And they can consume their body weight in tobacco foliage in a day, and every bite of that tobacco foliage contains nicotine. This is kind of a mind-boggling accomplishment, at least from the human perspective. We humans aren't really capable of eating our body weight in much of anything. I think the closest anyone probably has come to that is the world cranberry sauce eating title holder Juliet Lee, who's a 105-pound mother from Maryland who ate 13.23 pounds of cranberry sauce in 8 minutes, which was equivalent to 12.6% of her body weight in the process. Not a record that people are probably anxious to break. So, given the toxicological challenges that caterpillars, who are not particularly mobile, they don't have wings, they can't leave their host plant, they're pretty much stuck when their egg hatches on eating whatever they can find under their feet. Over 90% of lepidopterans, those are butterflies and the immature stages of butterflies and moths, are restricted to feeding on plants in 3 or fewer plant families. That is, they are specialists. They feed only on a very limited range of the 250,000 plus flowering plants in the world. And one consequence of being specialists is that they are extremely good at detoxifying a relatively restricted range of phytochemicals, those that are most frequently encountered in their host plants of choice. So both vertebrate and invertebrate organisms, including humans and caterpillars, depend on 3 major classes of detoxification genes to metabolize, solubilize, and transport toxins. The phase 1 enzymes, notable among which are the cytochrome P450s, basically change the structure of a toxin so it no longer can interact with a vulnerable site in the body. The phase 2 enzymes, including glutathione transferases, attach moieties to the metabolites to solubilize them and make them more transportable. And phase 3 enzymes and other gene products are responsible for basically escorting these conjugated metabolites out of the body. So humans and insects, along with most other aerobic organisms, rely very heavily on the cytochrome P450 monooxygenases for that phase 1 detoxification. The P450s owe their name to the fact that in the laboratory, the P450 enzymes display a different spectrum at 450 nanometers. When you expose them to carbon monoxide, they shift to 420. Not a particularly user-friendly name. They are membrane-bound heme proteins, a very diverse multi-gene family, over 11,000 sequence members, and they are structurally highly diversified, except they all, with a few exceptions, share a heme-binding signature motif. So they are oxidizing enzymes and depend on interacting with heme in order to have their catalytic activity. Now, there are 11,000 plus P450s, so as you might imagine, it's hard to come up with names for everything, so that many different enzymes. So there's a systematic way of naming them where they're all called by CYP, cytochrome P450, and if there is 40% or greater sequence identity in the amino acid composition, they are assigned a numerical family. 55% or greater identity leads to assignment of a letter to designate gene subfamily. Then within the subfamily there are genes, and if they're 98% or more identical at that particular locus, they are allelic variants. So the P450 monooxygenases are called multifunctional oxidases because they can handle a great many substrates, and they are capable of catalyzing a great many different types of oxidation reactions. So basically, they take a lipophilic, fat-soluble substrate and oxidize it so that it becomes more water-soluble, hydrophilic, and hence excretable. In the process, they split oxygen, basically oxidize the substrate, and produce water at the end of the reaction. So P450s in most organisms have 2 different types of substrates. There are endogenous substrates. Those are hormones and pheromones, and they are involved in the biosynthesis of these endogenous compounds that have a use, that are essential to the physiology of the organism producing those P450s. But a great many, in fact the majority of P450s, deal with exogenous substrates. So these are materials that are outside the body. They're present in the environment. They are xenobiotics, foreign substances, and to some degree signaling substances between individuals called pheromones. And the P450s are involved not in biosynthesis of these exogenous substrates, but their degradation and detoxification. Now, molecular analysis of these P450 sets the origin of the gene family about 400 million years ago, which happens to be coincident with the colonization of terrestrial environments by plants and their herbivores. So this is when ancestral living organisms crawled out of the primeval muck and encountered an aerobic atmosphere. Among other things, they also encountered fire and combustion products, which don't exist underwater, and they also, most importantly, encountered each other. So invertebrates climbed out of a mire, primeval mire, and encountered plants, and the interaction began as a consumptive relationship. So the suggestion has been made that reciprocal adaptive evolution has led to reciprocal radiations in the biosynthetic P450s in plants to produce chemicals to protect them against herbivores, and in the detoxification genes in insects to allow them to meet the toxic challenge presented by plants when they biosynthesize new and different types of defensive compounds or phytochemicals. Whether substrates of P450s are endogenous or exogenous influences the number of genes in each cytochrome P450 family. So plants make biosynthesize phytochemicals by P450s for defense. Those P450s tend to be diverse and idiosyncratically distributed, just as the insect P450s that are involved in detoxification are diverse and idiosyncratically distributed. By contrast, those with highly conserved endogenous substrates also tend to be highly conserved. So in this reciprocal adaptive evolution, sometimes called a coevolutionary arms race, there are multiple gene duplication events that allow these environmental response genes, so genes that have as their, that encode enzymes that have as their substrates exogenous compounds made by other organisms. Gene duplication events allow them to proliferate, diversify, and acquire new functions as new substrates are encountered. So by contrast, those genes, including P450s that are involved in endogenous functions such as steroid biosynthesis, remain highly conserved. In insects, the main endogenous substrates for P450s are two types of hormones that are involved in molting, which is kind of essential to the whole process of being an arthropod. Ecdysone is called molting hormone, and that is the hormone that allows insects and other arthropods who are trapped in their exoskeletons to get bigger. They have to grow, so they have to shed their exoskeleton. So molting hormone allows them to undergo molting. Juvenile hormone allows them to retain their immature status as they go from one stage to another. They don't become adults until juvenile hormone titers decline in immature development. So these are hormones that are found almost universally in the class Insecta, and the P450s involved in their biosynthesis are highly conserved. Just to see the difference between P450s that are involved in ecological interactions versus P450s involved in physiological processes, it's instructive to compare the cypomes, or the cytochrome P450 inventories, across multiple insect genomes. So Nasonia vitripennis is a parasitic wasp. Apis mellifera is the western honeybee. Anopheles gambii is the malaria mosquito. Drosophila melanogaster is the workhorse of genetics. It's called the fruit fly. Entomologists actually call it a vinegar or pomace fly, more correctly. And Tribolium castaneum, which is a red flower beetle, stored product pest. So each has a unique and distinctive cypome, and you can compare across what are called clades. So there are so many P450s. They are not only classified in families, but families that are grouped together in clans or clades. So looking at the clan level, you can compare the size of the different P450 groups, which should vary according to the ecology of the organisms. So CYP2 is a clan. And these are uniformly involved in endogenous functions, specifically in producing those molting hormones, the steroidal molting hormones. CYP307A1, for example, from Drosophila is one such CYP2 P450. So these are the CYPomes of the 5 representative genomes, and if you break it down and zoom in, you see that the CYP2 genes across these 5 genomes are small. They're low numbers. They are highly conserved sequences. They're highly conserved functions, and you can see orthologs clearly across these genomes. So with Drosophila in blue, you can see that each of the Drosophila CYP, here's Drosophila 307, has a clear ortholog in the malaria mosquito, in the parasitoid wasp, in the beetle, and back here in the honeybee. So it's easy to find the orthologs because they're conserved, they're working on the same substrate. Same thing for the mitochondrial clan. These also have endogenous functions. The mitochondrial P450s are also involved in many of the biosynthetic steps that are associated with production of the molting hormone, ecdysone. And similarly, these are across these 5 genomes. There's not that much variation in size, with a low of 4 in the parasitoid wasp and a high of 11 in the fruit fly. Low numbers, highly conserved sequences, and highly conserved function with very clear orthologs. Again, you can, for every Drosophila mitochondrial P450, you can pretty much see the ortholog in the other insect genomes. Everything is different for the CYP3 clan. The CYP3 clan is where most of the families that are involved in detoxification of exogenous substrates, those from the environment, are found. They have detoxification functions, and what you see in these genomes are what are called blooms, which are sort of explosive adaptive radiations, where all kinds of duplication events have allowed multiple variants to accumulate, diversify, acquire new functions. So typically the CYP3 clan P450s are characterized across genomes by high numbers, little sequence and functional conservation, and no clear orthologs. So here you see you see the color bursts here. So this is Apis mellifera, all very closely related to each other, but not really closely related to any other group of CYP3 P450s and any other genome. The size varies enormously as well, from a low of 28 CYP3 P450s in the honeybee to 70, almost 3 times as many in Tribolium castaneum, the red flower beetle. And CYP4 is a bit of a mixed bag, literally, because some of the CYP4s are known to have exogenous or environmental substrates. Others are involved in endogenous functions, some such as functioning in fatty acid metabolism as omega hydroxylases. Some are involved in pheromone degradation, and as you might expect, it's a mix. They're in between. Some of the CYP4 clan P450s are display these adaptive radiations, these blooms, whereas others, such as here's Apis mellifera 4G11, that is a group of CYP4 P450s where each genome has a clear ortholog. And not exactly clear yet. We're still trying to figure out what CYP4G11 is doing. Now, not everybody has embraced the idea that ecology, that these interactions, this coevolutionary arms race, can explain variation in the numbers of P450 genes in any given genome. This is a Setsutsu et al. In 2013, in fact, argued that patterns of gene duplication and death in the CYPome are not dependent on ecology and life history traits of the organism. Plant-animal chemical warfare, widely thought to be a root cause of the proliferation of CYP genes, does not shape their distribution into families. And as evidence, these authors compared the CYPome of Tetranicus urticae, which is a two-spotted spider mite, which has hundreds of plant family hosts. It is actually a major plant pest, feeding on hundreds of families of plants. By contrast, Bombyx mori, the domestic silkworm, has only one known host plant, and that's the white mulberry, Morus alba. And the same distribution of genes per SIP family appears to be the case, and it appears as if there is no room there. There's no signal of the differential diet breadth in this comparison. It's kind of not exactly a fair comparison in that Bombyx mori is pretty much a domesticated animal. It does not exist in nature. It couldn't survive in nature. You see here, it has no pigment. The males basically are not very good flyers. It feeds only on one host, and it's incredibly finicky at that. Notwithstanding, it certainly is restricted to a single host, but part of the problem in looking at the whole SIPome is the whole SIPome is not involved necessarily in the interactions between plants and insects. So as well, it's hard to define exactly what constitutes a prolificous species or a generalized feeder or a specialized feeder. In defining what's a specialist or a generalist, insects actually are more chemists than they are plant systematists. And just a case in point, their feeding habits often follow not necessarily plant taxonomy, but the distribution of particular phytochemical groups. Here is a group of phytochemicals that are that humans have interacted with quite a bit, sometimes with adverse consequences. They're called furanocoumarins. So this is a furan ring that is attached in the furanocoumarins to a coumarin nucleus, and in this linear, they're basically two flavors, as it were, a linear configuration where the attachment is 6-7, and the angular configuration where the attachment of the furan ring is 7-8. Linear furanocoumarins occur in about 10 families. Angular furanocoumarins are pretty much restricted to only 2. That's one of the hallmarks of phytochemicals. They are idiosyncratically distributed. They occur with diversity and abundance really in only 3 plant families: the Apiaceae, or the carrot family, the Rutaceae, the citrus family, and the Fabaceae, the legume family. They are highly biologically active because in the presence of ultraviolet light, they can form covalent links, bind to pyrimidine bases in DNA and cross-linking, and in opposing strands, and cross-link the DNA to interfere with transcription and replication, which is pretty toxic to anything with DNA, which is life on this planet as we know it. And humans, even contact with furanocoumarins, casual contact of plants that contain furanocoumarins in the presence of sunlight, produces this phenomenon called photophytodermatitis, sometimes called phytophotodermatitis, where the lesions form on the skin, blistery eruptions, weepy, oozing, painful, unsightly blisters. In this case, it's called Burlock dermatitis, and was discovered around the turn of the 20th century because people who went sunbathing, which was a new hobby in the beginning for women at least, in the beginning of the 20th century, when there's finally enough leisure time to enjoy swimming at the beach, would apply oil of bergamot to places where perfume goes, on the inside of the wrist and on the neck and behind the ears. Oil of bergamot is from Citrus bergamia, which is a rutaceous plant, the peel oil of which, the essential oil of which, contain furanocoumarins, and hence Burlock dermatitis. A more contemporary manifestation of furanocoumarin-associated phytophotodermatitis was called Club Med dermatitis, when a woman returned from a Club Med vacation with linear mirror image hyperpigmented patches with scallop borders on her mid-medial thighs. When the physician asked her what on earth they were doing on this Club Med vacation, she mentioned that there was a drinking game during which participants would balance and roll limes up and down their laps and closed and presumably naked thighs. And that's consistent with the fact that lime peel oils, again, limes are citrus in the citrus family, loaded with furanocoumarins. Mexican beer dermatitis which is another unique variant of Lyme phytophotodermatitis attributable to drinking practices. Scourge of the beach bum and the pauper lady. This is sometimes called bartender's disease because bartenders who make a lot of Mexican beer or gin and tonics in the summer end up with lime juice in the presence of sunlight. Now, there are phyranacumens in our diet. We eat them, but nobody eats plants that contain them with regularity. Here's a Ivy et al. From Science in 1981 reporting the discovery of, or the occurrence of phyranacumens in levels of parts per million in raw and cooked parsnip root. Suffice it to say, we don't eat a whole lot of parsnip roots. It's kind of pretty much relegated to side dishes and occasional appearance in soups and stews. We don't eat them in regularity, and we don't typically eat vegetables in quantity. And here's what happens when you do. This is a paper in Archives of Dermatology of a woman who made the mistake of eating, for reasons that were not disclosed in the paper, about 1 kilogram of celery root, drinking the water she had cooked it in, and then went to a tanning salon. And she ended up with third-degree burns and that were associated with exposure to phyranacumens in the parsnip root and the celery root. And the warning here, yeah, it makes you rethink celery, doesn't it? Vegetarians should be warned to avoid massive UVA exposure from outdoor sun or artificial sunbeds after the intake of large quantities of celery. Now, large quantities of phyranacumens are ingested by the black swallowtail, Papilio polixenes, which is restricted to basically 2 host plant families. Feeds only on plants in the Apiaceae and Rutaceae. Essentially, all host plants of the black swallowtail contain both linear and angular phyranacumens, although overwhelmingly linear phyranacumens. Wild parsnip, from which we get parsnip root, contains about 6 different compounds. Black swallowtails are far more tolerant of phyranacumens than our caterpillars that feed on a diversity of plant families that lack phyranacumens. Spodoptera eridania feeds on dozens of plant families. It's the southern armyworm. It avoids plants with phyranacumens with good reason, because phyranacumens, the xanthotoxin, linear phyranacumens, toxic to eridania at parts per billion levels, whereas black swallowtails can tolerate them at the parts per thousand levels. Now, swallowtail, in the family Papilionidae, the swallowtail family, phyranacumene feeding occurs only in one subfamily, the Papilioninae, and only in one tribe, the Papilioninae. And not even all members of that tribe contain, eat plants containing phyranacumens. Some of these caterpillars have host plants, all of which contain phyranacumens. Some only occasionally encounter them, and then particularly in other tribes within the subfamily Papilioninae, never encounter them. Their ability to metabolize them and to upregulate the genes encoding the enzymes, so to bulk up the enzyme activity in the presence of ingested phyranacumens, typically is found only in the caterpillars who always encounter phyranacumens or occasionally encounter them. And in fact, those that encounter them only occasionally have lower levels of constitutive induced levels of activity. By contrast, those species that never encounter them cannot metabolize them at all or tolerate them. So in 1992, working with Mary Schuler, who was at the time a plant P450 investigator, we isolated the second plant insect P450 known, so CYP6B1V1. So CYP6 is an insect P450 family. The first one was CYP6A, that was one which was identified from housefly, even though CYP6B1 shared only 32% identity with the housefly P450, because the vast majority of P450s at the time were vertebrates. It was lumped together with the housefly in family 6, different subfamily. And as it turns out, unlike human P450s that tend to have very broad substrate specificity, as befits our very broad diet, biochemists call this kind of activity promiscuous, which I think is like slut-shaming enzymes, but we won't go there. The black swallowtail P450, CYP6B1V1, is actually pretty selective. In fact, differentially has high activity for the linear phyranacumens and not so much for the angular phyranacumens. we found a second P450, CYP6B3, in Papilio polixenes several years later, and wondered whether maybe that was the backup P450 in order to enable the black swallowtail to tolerate even low levels of angular phenocoumarins in its host plant. Eventually, heterologously expressed it. This is a paralog of CYP6B1 resulting from a recent gene duplication event. CYP6B3 has more allelic variants, more single nucleotide polymorphisms, more non-synonymous substitutions, those that affect amino acid sequence, particularly in substrate recognition sites. So it has the hallmark of selection to differentiate on its function. So we heterologously expressed it. We were surprised that uniformly CYP6B1 in its allelic variant is far superior in metabolizing linear phenocoumarins than is CYP6B3 in either one of two allelic variants. It's not even very good at angular phenocoumarins. In fact, CYP6B1 is 10 times better at both linear and angular phenocoumarins. But what CYP6B3 is good at is another group of phytochemicals called pheranochromones, which are found in some of the apiaceous hosts. So this gene duplication event that led to a paralog conveyed upon swallowtails the ability to colonize phytochemically different plants within the family Apiaceae. So this is a gene duplication event led to the evolution of both differential regulation and complementary functions. Not all Papilio species are phenocoumarin specialists. Papilio glaucus group, this is a group of tree-feeding swallowtails. They are considered prolificous. They feed on about a half a dozen plant families rather than just Apiaceae and Rutaceae. So this is the eastern tiger family. Swallowtail feeds on the tulip, the Magnoliaceae, the magnolia family, the ash family, and the wild cherry family. In contrast, Papilio canadensis, the Canadian tiger swallowtail, has no phenocoumarin-containing hosts. It lacks that rutaceous host, Bacteria trifoliata, that's called the hop tree. So while the tiger swallowtail, eastern tiger, has a single phenocoumarin-containing host, the Canadian tiger has none. If you compare their P450 SIP-homes, CYP6B P450s have two distinct complexes in these two species, CYP6B17 and CYP6B4, and identities range between 93% to 95%. And functions are dramatically different. Tiger swallowtail P450s have much lower activity, but broader substrate specificity than CYP6B1 in the specialist black swallowtail. Black swallowtails are very, very good at linear phenocoumarins, which they ingest with regularity and at high concentrations, not so good at angular phenocoumarins. The CYP6B P450s in the tiger swallowtail, eastern tiger, PG here, have a broader range of substrates and lower activity. Papilio canadensis, which never encounters phenocoumarins, is not very good at metabolizing any of them. So multicaudatus, the two-tailed swallowtail, is putatively ancestral to this prolificus group, the glaucus group, and it does include among its three host plant families hop tree, Telia trifoliata, which contains phenocoumarins. So here it is. Phenocoumarin feeding evolved early in the evolution of the genus Papilio. It's widespread in the genus. It's lost here in the glaucus group, right here, although it's present in the ancestral multicaudatus. And there are an intermediate number of P450 CYP6Bs in this intermediate semi-specialist versus the generalist. And in fact, CYP6B33, which is a phenocoumarin metabolizing P450 in multicaudatus, is intermediate in activity and intermediate in its distance to the heme, that reactive site, which determines its activity. So CYP6B1 is the enzyme is very well constructed to metabolize efficiently the phenocoumarins. The glaucus CYP6B4 is a more generalized enzyme, can handle more substrates farther away from the heme group, and CYP6B33 is in the middle. Now, lepidopterans other than swallowtails eat phytochemical-containing plants, like vertebrates with our promiscuous P450s, which is appropriate given the breadth of the diet of most vertebrates, in truly prolific insects like the corn earworm, which is also known as the tomato fruit worm, cotton bollworm, and false tobacco budworm. P450s are also promiscuous, such as CYP6B8. So here you see CYP6B8 can metabolize not only natural products such as flavone and rutin, but also synthetic insecticides. So that's CYP6B8. By contrast, CYP6B1 has virtually only one set of substrates, the phytochemicals. CYP6B8 and other corn earworm enzymes are also upregulated by a diversity of natural and synthetic substrates. The ultimate example, the parsnip webworm, Deiphoceria pasnicella, is a super specialist. It ingests only it feeds on only two genera of apiaceous plants, parsnips and cow parsnip, Pasnaca and Heracleum. It ingests the equivalent of its body weight in parsnip flowers in a day, so it can consume up to 10% of its body weight in toxic phytochemicals every day. Unlike the swallowtail that feeds on foliage, which have low levels, the parsnip webworm feeds on flowers and fruits that have the highest levels. And it has extremely specialized P450s. CYP6AB3 in the parsnip webworm is astonishingly substrate specific. It metabolizes only one of the 6 phytochemicals that you find in parsnip flowers. That's impertorin. So that's one way to eat a plant, but back in the Cretaceous, a new type of herbivore appeared, and that was a mutualistic pollinator. So bees and flies and butterflies and moths evolved to consume a new kind of plant tissue, that is nectar. Nectar evolved solely as a reward for mutualistic partners. And it's mostly sugar, but it contains phytochemicals as well. Now, bees are herbivores that diversified as specialists on pollen and nectar. Bee genomes are deficient in genes relative to non-bee genomes across all of the clans, especially the detoxification clan, clan 3, which includes family 6 and CYP9, which are associated with detoxification. So here is a hymenopteran, a wasp. It's a solitary parasite, and it contains 92 P450s. By contrast, Apis mellifera, the western honeybee, and Bombus terrestris, the bumblebee, have about half that number of P450s total. But it's not uniform across the family. Remember, families are not the level at which selection is probably taking place. In comparing the parasitic wasp with the pollinator, you see that the pollinator in black has more CYP6A subfamily P450s than does the wasp. And these CYP6A P450s are upregulated by specific chemicals that are found in honey, pollen, and propolis, which is a sort of a bee glue that bees make from resins they collect from plants. So these are specifically upregulated by compounds found, phytochemicals that are characteristic of nectar and pollen. So Wen-Fu Mao, my postdoc, constructed a molecular model of these CYP6A enzymes, which allowed us to use in silico high-throughput docking to look at 14,500 compounds at once. Of the top-ranked 26 compounds, 6 were flavonoids, and particularly flavonols. And that's what these enzymes are very good at metabolizing. They're, in fact, way better at metabolizing flavonols than even the very generalized caterpillar, the corn earworm, which encounters flavonols in foliage. Well, they're, at least they encounter lower concentrations. The corn earworm is better at metabolizing them, but has fewer enzymes that do. So bees don't eat nectar, they eat honey, and honey is made of nectar from flowers. Bees collect a whole lot of different nectars. A single bee can make 30 foraging trips in a single day, visiting 50 to 100 flowers at a time, brings back the nectar to the hive, transfers the nectar to a worker in the hive who sucks it up, spits it out 100 to 200 times to evaporate it down to about 18% water from 80% water. And after processing, it's capped into wax cells, it loses water, and in addition, So in addition to forming a supersaturated solution of sugars to make honey, bees also make a concentrated solution of nectar phytochemicals, which are largely flavonoids and phenolic acids. So before the honey is capped, it sits in the hive for a few days, and at hive temperatures, some of the toxic nectar constituents, including some phenolics, actually break down. So bees essentially invented cooking long before we did, letting the honey cure at hive temperatures of 95 degrees or greater allows them to cut down a little on the phenolic content of their food. And pollen is also hard to digest. Bees don't actually digest pollen on their own. They eat bee bread for the most part, a combination of pollen grains, nectar, glandular secretions, and gut fluids, which is similarly capped and ripened in the hive for about 2 weeks. So there's a fermentative process with mutualistic fungi. The bee bread chemistry changes, but what is released includes these flavonols. Every pollen grain contains flavonols, particularly camphorol and quercetin, which are signaling compounds for the pollen. And they're in every pollen grain. Propolis, which bees gather to seal the cracks in their hive and also to confer antibiotic activity, these are rich in flavonols as well. And no matter what plants are used for making propolis, and it varies all around the world, flavonols are a recurring theme. So just about everything bees eat, including pollen, honey, propolis, contain large amounts of quercetin or closely related flavonols. Honeybees lead a flavonoid-saturated life. In honeybees, quercetin upregulates all kinds of these CYP6A-S as well as CYP9, P450s, all of which metabolize these quercetin and other flavonols. Why so many? Well, we showed by RNA-seq analysis that larvae in their first 3 days that are given quercetin experience downregulation of the energy-generating pathways in their mitochondria. Why does that matter? Well, quercetin then can power down the powerhouse of the cell. Mitochondria is where cellular fuel comes from. ATPs are made. So flavonoid detoxification is utterly essential to the bee lifestyle. What about other bees that subsist on nectar and pollen? Well, there's an array of solitary to social living styles across bees. So some, like Apis mellifera, the honeybee, and the dwarf bee, honeybee, live in large colonies year-round. They're perennially eusocial. Bumblebees live in, so honeybees have 30 to 50,000 members. Annual bumblebee colonies, only the queen overwinters, and then during the year, colonies build up to 300 to 500. There are others that are kind of mixed, partly eusocial, partly solitary. And then a number of bees, such as the alfalfa leafcutter, live a solitary existence where a female basically feeds pollen to her own offspring and not the queen. So eusocial bombus and aphis. So bumblebees and honeybees have a reduced inventory of P450s relative to other eusocial insects such as ants, but the CYP3 clan is basically the same size in bumblebees and honeybees. So at the CYP3 clan, or even the family level, there don't seem to be differences in P450 diversity that relate to sociality, but at the subfamily level, what happens is that clearly there's a proliferation, an increase in the CYP6A-S, the flavonoid processing P450s. And in fact, the degree of sociality that is displayed in this continuum of solitary to social across all kinds of bee genomes is significantly correlated with the size of the CYP6A-S inventory. So bees are basically specialists on flavonoid-containing plant materials, nectar and pollen. They differ from virtually all other pollinators by virtue of consuming nothing but pollen and nectar during all life stages. Perennial eusocial bees differ from other bees by virtue of concentrating nectar into honey and converting pollen into bee bread. Perennial eusocial bees also differ from other bees because they gather these plant resins to protect their hive resources from microbial contamination and weather. Other than pollen wasps, a very obscure group of wasps called masarine wasps, maybe the wax moth, which is a caterpillar that lives in beehives, and a pollen beetle that also eats just pollen. Virtually every other insect that consumes pollen and nectar feeds on other foods during some part of its life cycle, such as this pollinating hoverfly, which as a maggot feeds on rotting plant debris. So in addition to the evolutionary progression from solitary to perennial eusocial lifestyles, there's an evolutionary progression from floral resource specialists to floral resource super specialists, i.e. from short-term storage of ephemeral low-quality foods to improving and stockpiling those foods. Along with the degree of sociality, there's a change from short-term storage of ephemeral low-quality foods to processing, improving upon, and stockpiling those foods. And that's what we do. So we have, as evidence, our little Debbie honey buns, perhaps not as nutritious, but bees and people are similar in many surprising ways. We are gleaners. We also feed on a wide range of plant materials. We consume a lot of flavonoids, and we also communicate with each other. Now, the downside of a broad diet is it's susceptible to interactions. In humans, this risk is manifested in the form of phytochemical drug interactions. Those furanocoumarins that you find in citrus plants like Rutaceae, like grapefruit, are suicide substrates for P450s. And that's why so many drugs have warnings on their labels. You should not drink grapefruit juice when you take medication, because the grapefruit juice furanocoumarins will block your P450s from detoxifying the drugs you ingest, and you'll experience essentially an overdose. Bees may be especially similar to synergistic interactions among P450 substrates because they have so few xenobiotic metabolizing P450s. And in contemporary agroecosystems, they encounter new dietary toxins at an accelerating pace, not only in natural toxins, but also non-hive toxins that are used to control mites and other parasites inside the hive, and then agricultural toxins. Pesticides have started to show up with regularity in the hive, including every class of pesticide, in the acaricides, herbicides, especially fungicides. And 121 different pesticides and metabolites have been found in wax, pollen, bee, and hive samples. 98% were contaminated with an average of 6 pesticide detections per sample and a high of 39. Wax is incredibly good at absorbing these lipophilic pesticides. So 9Q1, which is one of the P450s honeybees rely on to detoxify flavonoids and other toxins, when you do an in silico docking, lots of insecticides, fungicides, are known inhibitors of P450s, dock into the catalytic site and can interfere with the ability of honeybees not only to detoxify pesticides, but their own food. When adult workers are fed quercetin, the flavanol, in the presence of a fungicide, myclobutanil, quercetin accumulates in the hemolymph, and the amount of ATP present declines in thoracic muscles. They need ATP in their thoracic muscles to fly. So fungicides may not be acutely toxic to bees directly, but they can keep the bees from metabolizing the phytochemicals present in their food. Now, we consume P450 inhibiting pesticides too. Do they compromise our ability to detoxify phytochemicals? Another reason, perhaps, to rethink celery, which is one of the most heavily pesticide-contaminated vegetables that we consume. And remember, it's got those furanocoumarins in it, not to mention vegetable Viagra. So thank you. Thanks to the National Science Foundation, the USDA for funding, the many, many teachers, mentors, students, collaborators, and friends who made this work happen.